Hi, my name is Eric, and today I'm going to talk to you a little bit about what we do at my work, which is an organization called AdGene. AdGene is a plasmid repository. So to understand what that means, we have to learn what a plasmid is. And to teach you a little bit about that is a song that we made a few years ago with some kids who are a little bit older than you, but I thought you might like to watch because it's a fun way to learn about what plasmids are.
A plasmid is a circle of DNA with lots of parts. Bacteria make them, but scientists can't break them apart. So now I'm so glad that we can design them a la carte. So they should be shared, be shared, be shared. Plasmids turn genetics into art.
Thinking about playing with genes. What if we could change them and spread it like memes? What if we could rearrange them to study proteins? Well, these plasmids got us all letting off steam. It's lit. So here's what a plasmid is. Circle of DNA from bacteria. It's got restriction sites where we slash with scissors. Called restriction enzyme and let us match a mix. So mix and match genes from a new species. Just put in a promoter and express with ease. And with a site of origin, it can replicate. Make copies of your plasmid and generate. Yeah, they're great. Easy to manipulate, but just think, what if they could infiltrate not just bacteria but other life? We'd be pretty much wielding a genetic knife.
A plasmid is a circle of DNA with lots of parts. Bacteria make them, but scientists can't break them apart. So now I'm so glad that we can design them a la carte. So they should be shared, be shared, be shared. Plasmids turn genetics into art.
Okay, snip, poke, test, cells, pipette, run, gels, ligate, incubate, die, wait, survive? Great. Now each ring has a power like Frodo done told me. And I got so many you could call me Kobe. Favorite application is to make them glowing, which helps you find out where molecules are going. All I do is insert GFP in the right type of ring with the right set of genes. Depending how I do it, it'll be glowing green or blue, either all the time or when I want it to. Like maybe just in cells of a certain type or biosensor for molecules floating by. And I could do this in fish, I could do this in a beagle, I could do it to you, but that'd be illegal. But we do it to research, to state in nature, golden age of biotech and its application. From medicine to food to security, to learn about the fundamentals of biology.
Plasmids, what new genes can we add in? What new questions we asking? Plasmids, plasmids. Plasmids, what new genes can we add in? What new questions we asking? Plasmids, plasmids.
Hey everybody, I'm Tom. Thank you so much for watching, and huge thanks to
All right, so that's a little bit about what a plasmid is. So basically, it's a circle of DNA. And let's see. So DNA, as you've learned from, you may have seen from other videos, is something that makes up all of us. And AdGene is, we call it a repository, but really what that means is essentially we're kind of a library for DNA. So instead of, at a library, you go and you get books and on bookshelves, and at AdGene, we put our plasmids, our DNA, into freezers. So this is the inside of our lab. And we've got lots of these very, very, very cold freezers that are about minus 80 degrees centigrade. So very cold. So where does the DNA come from that are in these plasmids? What kind of genes do we have? We have them from lots of different organisms. We have them from bacteria. This is a blown up picture of E. coli, which is a very common form of bacteria that we have. And we have them from other types of disease-causing things like virus. This is a picture of coronavirus. So we actually have genes from the coronavirus. And scientists from around the world ordered those genes from us when they were studying how to create vaccines and other things for the pandemic. We also have genes from different types of foods. So we have genes from corn, from peach, lots of different types of plants. And then we also have genes from some of the animals that people eat, like cows, pigs, and chickens. And then we have genes from what are called model organisms. Model organisms are types of creatures that people study in order to look at human disease or agriculture or any number of things. So even though they look very different from people, people still use yeast. So this is a picture of yeast. Small worms called C. elegans. Fruit flies are a very common model organism. Zebrafish and mice are also very common. And believe it or not, we even have genes from human beings. So lots of different genes from human beings. Not every gene, but some, so that people can study the diseases and that humans get. Some other interesting genes that we have can be used for as special tools. So we have genes such as the gene that makes fireflies glow, and also genes that make jellyfish glow. And these are really cool. Scientists use these all the time to make different things glow inside our cells and inside different types of organisms. So here's a quick demonstration of what some of these things can do. So this is a plasmid, which has a type of green fluorescent protein in it. And the green fluorescent protein comes from a jellyfish. So there's the jellyfish, and this gene helps jellyfish glow. And we can take that gene and we can put it into bacteria, in this case, E. coli. Now, we don't use the E. coli that make people sick. We use it for other things. But this is a quick video of some of my colleagues doing something very special with some bacteria that have been transformed with this special plasmid that makes them different colors. So this is just to show what they're doing. So
I'm sorry, I want to change my blog.
I am talking in this video, but it's a little quiet. So I'm going to mute it and I'm going to talk right now. So basically what my colleagues Dolly and Uma are doing is they've taken these agar plates, and agar is basically bacteria food. And they have these special bacteria that have these plasmids in them with different genes that make them different colors. And they can take toothpicks, stick them into a little tube of bacteria, and then spread them on the plates. And so that's what they're doing here. And it's kind of like painting, except you can't see anything right now, because when they first put that on there, the bacteria are invisible. And I don't know if you can guess what they're doing based on the letters that are on there, but I'm going to stop the video. So what they're going to do is they put that on there, and then they put them in what's called an incubator. The incubator grows the bacteria overnight. And then the next day, we get this. So there's our school name, Butler. And this is what E. coli look like when they don't have plasmids in it, but we can put those special genes in them that make them different colors. So we have some that glow, that are bright pink, and then we have some that are bright green. And we can do this to all sorts of organisms. You can put these genes into mice, you can put it into plants, you can put it into different things. And that's how scientists use plasmids to study different organisms and learn more about them. So thanks for learning a little bit about where I work.